Hi, my name is Rashik Amin. Today I will be explaining the process of DNA replication, transcription, and translation. First, I made a poster board explaining the processes. Here's the nucleus, and here's the cytoplasm of a cell. Replication is a process that produces two identical DNA replicas from one original DNA molecule. Transcription over here is the process where a particular segment of DNA is copied into RNA. Translation, on the other hand, is the synthesis of a protein that is directed by a mRNA template. I will first explain the process of DNA replication. We start with a molecule of DNA here, shown here, which is a helical structure. The enzyme helicase comes and breaks the hydrogen bonds between the nitrogenous bases. Here you see that the helical structure is breaking down. DNA polymerase III is responsible for the actual replication. DNA is always synthesized from the 5' end to the 3' end. This means that DNA polymerase will be moving from the 3' to the 5'. There's a leading strand shown here and a lagging strand here. The lagging strand is synthesized in chunks where RNA primers were placed. The leading strand is being synthesized from 5 to 3. DNA polymerase I removes the RNA primers and replaces them with DNA. Finally, ligase comes in and fixes all the phosphodiester bonds, and replication is complete. Next, I'll be talking about transcription down here. RNA polymerase uses one DNA strand as a template to make mRNA. Exons stay in and introns are spliced out. Here you can see the complete mature mRNA with a poly A tail on one end and a cap on the other. Here's the poly A tail right here. The mature mRNA will leave the nucleus, and I drew it again right here. And this acts as a template to make a protein. tRNA is also made in the nucleus, and I drew it right here, and it's shaped like a T, as you can see right here. And it leaves the nucleus to go into the cytoplasm as well. Amino acids are shown here by the use of Skittles, and these are attached to the tRNA using tRNA synthetase, and I'll attach one right here to show you. rRNA is formed in the nucleolus, which is right here. And rRNA makes the small and large subunit of a ribosome. These will come out of the nucleus and attach to the cap end of the mRNA. The ribosome moves toward the poly A end of the mRNA, and it'll attach tRNA with mRNA, which is showed right here. The codons of the mRNA bind with anticodons of the tRNA when a ribosome is present. When this happens, the amino acid from the tRNA is released, and I'll show one released right here. The amino acid is released, and this amino acid joins with other amino acids from other tRNAs to form a polypeptide or a protein. This concludes the process of translation. Thank you.